think we'll get started again. Oh, I said that we would take some questions. Any questions over break, or we'll just start? Yeah? Mm-hmm. Yeah, so the question is, how sensitive is EB-Seq to input? Like, how good your expression estimates are? And the answer is, it depends, for sure, and power is the big issue. So if we have super, I won't say super, but as the expression estimation uncertainty increases, the power of EB-Seq decreases. We don't normally see a bunch of false discovery rates. Our false discovery rate is pretty well controlled, but if we have noisy estimates going in, we just don't see as much. We have I think one example in our vignette, which I'm going to try to show you some lines of code after this break. There's been a quick request for code, so we'll try to turn that around. And I won't go through the whole vignette, but at the bottom of that vignette, there's an example where we actually apply EB-Seq to data where we don't know the transcriptome, and so we do de novo transcript detection and expression estimation, and you can see that far fewer genes are found, isoforms are found. Yeah. Okay, so like EB-Seq and like much of the work I'm going to talk about today, this extension of EB-Seq to ordered experiments. You can think about them as time course experiments, although in the example that we first started working on, the conditions weren't ordered according to time. This extension too was motivated by the Thomson lab. So the Thomson lab studies this very interesting creature called the axolotl. And if you haven't met an axolotl, you should, I guess. They're pretty cool. And one reason they're so cool is because they have almost complete regenerative abilities. So you cut an axolotl's arm off, it doesn't seem to mind that much. It just grows one back. And it does so, throughout its lifetime. It doesn't lose this ability as it ages. So this is incredible, super interesting. This guy has a super huge genome, hasn't been sequenced, so when we look at RNA-Seq from this genome, this is one of our examples where we don't have an annotation for isoforms. Now this is in contrast to the mouse that we know really well, that has almost no regenerative potential. But what's interesting, right, is many of the genes that are known to be involved in regeneration in axolotl are present in mouse. They're active during development, and then they go to sleep. Right. So what's going on during development? How do the gene expression patterns between mouse during development compare with axolotl during development and regeneration? We're interested in those types of questions. So experiments have been done and continue to be done where we look at gene expression patterns in 7 positions along axolotl limb and along the mouse limb. So you can imagine, from proximal to distal, the arm is cut off in various spots, and then gene expression is measured in those positions. And so we want to compare these two. Are they the same as an axolotl? And so this is what motivated what we call EB-Seq HMM. There's a hidden Markov model that we build into EB-Seq. Okay, so here's what it looks like. Here's what the data look like. So we have genes, again, in the rows, and now we have multiple conditions. For a standard experiment, we'd just have 2. Maybe for a different type of experiment, we'd have 3, but they'd be in no particular order, whereas here we're imagining that these conditions are ordered, maybe proximal to distal, maybe early time to late time, whatever. And we find it useful to characterize the options of expression over these conditions according to 3 types. We call them paths. So there's a constant path where there's no change across the conditions. There's what we call a dynamic path, where every single transition we see a change. So here, up, 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 down, down, continuously up in this case. Dynamic paths, we see a change the whole time. And sporadic paths are a mixture of the two. For some transitions, we see no change, and for some transitions, we see a change. So 3 types of paths. And I think about these things Well, let me, if you know what HMM is, and if this doesn't make sense, that's fine. The transition here is what I'm actually going to call a state. So at the transition, I can stay the same, I can go up, or I can go down. Those are my states in the HMM. So let me say a little bit more about that. So HMM in general, what does it look like? It's an expression pattern process, so I have a bunch of data, a bunch of counts in different conditions, and they're ordered again. And I want to write down a model, or what I'll call a joint probability distribution for that data. Okay, so I need two things. I need states. I need to know the underlying states, what I'm going to talk about. I need a probability transition matrix that tells me the probability of going from one state to another at a given time, or at a given, I'm just going to call these time even though they're not ordered over time. So at a given time, what's the probability of moving up given that I moved up at the last transition? What's the probability of moving down given that I moved up at the last transition? Whatever your states are, and again, mine are no change up, down, I need to know the probability of moving from one state to another at time t, between time t and t minus 1. And I also need an initial probability distribution. So what's the probability that I'm in a given transition at the initial state? Now, once I have those things, I'm going to make a couple assumptions again, and I like to make those explicit. So I'm saying the observed expression vector is characterized by some model, and I'm going to use the marginal predictive distribution that we use in EB-Seq. So I use a beta negative binomial model, although you can change that. And here's the big assumption for the HMM that we make, and that everybody makes in an HMM, at least if it's first order, is that the joint probability distribution, given all the states, depends on only the previous state, and it can be written as a product over these states, right? So this is, if you think about your independence assumption, or the data at time t conditional on everybody else only depends on the previous times. Okay, so given that setup, again, our hidden states, that was sort of the HMM in general, our hidden states are up, down, and no change. They're states on the transitions. It's a little bit different than a typical HMM. And what we want to know is what's this configuration? That's what I want to know at the end of the day for a given gene. What's the gene's path? Did it stay the same? Did it go up, down, up, whatever? This is my object of interest. So again, in the dynamic, I have dynamic, sporadic, and then constant change is just I don't need an HMM for that. So in the dynamic case, what I have are these states moving over time, and I have an autoregressive component that describes dependence among the counts from time t to from time t minus 1 to time t. Okay. In the sporadic case, I don't assume that there's dependence among the states because things can be moving up, down, or no change. In the dynamic case, there's a hidden Markov component, this AR hidden Markov component. But here, there's a non-Markov component. And because I don't know which case I'm in, I have a mixture of these two. The emissions distribution looks a lot like it used to look with the beta negative binomial, except you'll see, and this is why I pointed this out to you before, we have an extra term here called Cassie. So this is exactly what we saw before, this R beta over alpha minus 1 that determines the mean, except here in the HMM case we have this Cassie value where it equals C if this transition is up, it equals 1 over C if the transition is down, and it equals 1 if it's EE. So that's how the means change in this beta negative binomial over time. Okay. So like EB-Seq, we do a bunch of simulations, and in any simulations we try to match features of the data. We work pretty hard to do that. We want the sample sizes to be comparable, the number of genes or isoforms to be comparable, the means and variances to be comparable, and so on. And we simulate 15% of the genes to be dynamic genes, so consistent change with what I'll call a strong signal, and then 15% with a weak signal, right? And 15% sporadic genes, same thing. 15% with strong signal, 30% sporadic genes, 15% are with a strong signal, and 15% are with a weak signal. Then 40% of the genes are just constant. And then we can evaluate EB-Seq HMM, DE-Seq2, which is out now, and EDGE-R. And what we see is that the power for the strong signals is comparable, about the same. But because we model dependence over time, we see a power advantage for the weak stuff. So you might not see, if you just look at two conditions or you look at all the conditions but you don't model dependence over time, for things that are weak, they're just changing slightly, we wouldn't know that they're differentially expressed. But because we model that dependence over time, we can find an increase in power for those weak signals. In addition, an advantage of this is you get the characterization of the paths. So two slight advantages. One, when you have stuff that's changing consistently but weakly, you have slightly increased power to find that with EBSeq, but I think more importantly is you have a characterization of the path. So with DE-seq2 or EdgeR, you can say, yeah, something's different here a lot of the time, but you don't know where the differences are, and you're kind of hard-pressed to give them a name, call them up, 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 down, or whatever, whereas here, you get a characterization of the path. Okay, so here's some genes identified exclusively by EBSeq HMM. In the simulation, and then a couple that we're interested in the actual case study. They like these two genes. And you can see the characterization was down, down, up, down, down, down. So down, down, up, down, down, down. That's the kind of output that you get from this. Now, part of the reason I like this output is because it's useful downstream, right? You can start clustering these genes by their expression patterns. We're very interested in Hox genes in development, and so we can characterize or cluster these Hox genes according to their up-down patterns. All right, that's a EBSeq HMM. It's a nice approach. It's not hugely different than EBSeq, so I'm not going to say a lot more about it. We'll talk about single-cell RNA-Seq next, unless there's any questions about the time course in particular. So again, the main advantage of the time course, EBSeq already allows you to calculate posterior probabilities of difference across multiple conditions, right? But when the conditions are ordered, if you use EBSeq HMM, you have slightly increased power, and then you can get, characterization of each gene or isoform according to these types of paths. So not a huge difference. Yes? Yeah, that's a great question. So how do you include covariates? And the answer is it's not easy in EBSeq. The way that we do it is we regress them out, and then we use the residuals on EBSeq. So if we have age, let's say, as a covariate, we regress out age, and we put the residuals through EBSeq. Our code is not set up, and our methods aren't set up to include covariates directly. It's a major weakness, but what we found is that by regressing out, we don't lose anything. And so it's kind of a quick fix that works really well. I do think it's a weakness, though, because people like to put in their covariates, and we just don't let them. Yeah. Yeah, so that's a great point. Is C specified by the user? You have an option to specify it, but we estimate it by maximum likelihood. Yeah. That's right. It's constant across transitions. Yeah. You can specify it not to be. Like if you want to see transitions that are big early on and then diminish, you can provide a C vector if you'd like in the code, but we estimate it. And when we estimate it, we assume it's constant across conditions. Mm-hmm.
Could you elaborate a little bit more on defining that difference between dynamic and sporadic? Like what constitutes being sporadic versus being dynamic in sort of a weird way? Does that make sense?
Yeah. So this question is can you talk a little bit more about the difference between dynamic and sporadic? So the difference, there's a difference in words and there's a difference in the formula. The difference in words is that for the dynamic paths, at every single transition, you are not in the EE phase. So every single transition is either up or down. The one I'm showing you here, it's all up, but it could be up, 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 down, down, down. Sporadic, for some of the transitions, there's no change. Okay, and then for some of the transitions, there is a change. What happens in the formula is that the transition between the states is governed by a hidden Markov model for the dynamic paths, but not the sporadic paths. So that's the main difference. Does that make sense? Yeah. Other things? Okay. So again, I'll try to get some code going over the break, and we can just briefly review code, look at the input, output. But we'll talk about single cell first, because it'd be weird to have a break after 10 minutes. Okay. All right, single cell. We're excited about single cell. If you haven't seen this, you'll be excited about single cell stuff. This is the December 2014 issue, I think, maybe 2013, of Science that talks about all the great stuff that single cell is going to bring us. I was super excited about single cell RNA-Seq because I had been looking at heat maps like this for 10 years, right? So here we have genes, and here we have conditions, and we have low and high expression, and this is just a study that I was involved in looking at diabetes and obesity in the mouse. The conditions here aren't super important. I can tell you about them if you care. But what I just want to stress here is that we've all seen these plots where, oh, these genes are, highly expressed here and lowly expressed here. And every one of those cells, as we know, cells meaning entries in this matrix, is average expression over 10,000 or more biological cells, right? So we can't see what's going on individually. So we're kind of always looking at X bar, right? But now with single cell RNA-Seq, we get to look at XI that makes up X bar. So that's awesome, right? I don't know if anybody else is as excited, but like getting to see the individual X's makes me really excited. So here's some individual X's. And we're going to notice some features of this data in just a moment. So here what I'm showing you is some single-cell RNA-Seq from human embryonic stem cells, the H1 line. We have genes here and samples on the columns as usual, and compared with bulk RNA-Seq of the same types of cells. So I'll say that white here are zeros. White here are zeros. So what do we notice? I'm showing you, I'd love to show you a heat map of all the data. Of course, we can't look at all the data real easily in one big heat map. So I'm showing you the 60th to 70th percentile of median normalized RSEM expression. So you can imagine one huge heat map where I've ordered the genes according to median expression, and I've just pulled out the 60th to 70th percentile, and that's what you're looking at. So what do we notice about single cell data compared to bulk? Yeah, not as much of a pattern. It's more heterogeneous, more variability, more zeros. Okay, more zeros. That's a huge issue, huge issue. Okay. So that's what you guys have already noticed. I don't even need to be here. You have more zeros. There's increased variability. And there's also multimodality, which is hard to see from the plot that I just showed you. So here's just a plot. The pink stuff is single cell data. The darker pink are just single cell. The variance is calculated across all the samples. And here we've removed the zeros. Okay, so this is non-zero in the lighter ones. And then the green comes from bulk. And I have collaborator data out here, and I have publicly available data as well. You can't really read the labels, although you'll have the slides soon and you'll be able to see the labels. The point here is that the variance is increased in single cell data.
The slides are already posted.
So the slides are posted. Okay. So that's one feature that we see here. So that was on the variability. Here's multimodality. So what we did here is we took every gene and we fit N plus to it. And we have about 90 cells in the particular study that I'm showing you. So we have pretty good sample size to identify multimodality. Yeah?
So that previous slide wasn't a surprise, right?
No, it's not a surprise. So the previous slide is not a surprise. Why? Because we know that the variance on X bar is, smaller than the variance on X, and we can write out the particular way in which it's smaller. So it wasn't a surprise, but it's nice to see it, right? There are some surprises, which I'll tell you later. Okay. But that wasn't one of them. We would expect increased variability because we've been looking at averages the whole time. So here we fit M-clust. It's just a very standard and good approach for identifying clusters in data. And we fit it to every gene, and then we record, because M-clust provides this for you, the number of modes that M-clust finds, the number of clusters that M-clust finds. And you can see for the bulk RNA-Seq, for most of these, it finds just a single. We see unimodality per gene. For a couple of them, there's multimodality, right? But for single cell, there's increased multimodality. So people know what I'm talking about. I'm talking about the expression of data within gene. We made assumptions back with bulk RNA-Seq that that expression looked multimodal, right? I said conditional on a given latent level of expression. I have a distribution that describes biological and technical variability. And the distribution I used was negative binomial, and it's unimodal, right? For single cell, we better be worried about that distributional assumption if we make it. Right, we better be worried about it because for a lot of these genes, I see two modes. And this is within one condition. This is not due to differential expression. So I need to think about how to model that data. Okay, so we are working on, I would say, 3 main problems in single cell right now. And I'm only going to talk about one of them, but I'll briefly step aside to mention the other 2. The first one is normalization. Normalization is a big issue with single cell data. If you look at the first 10, 15 papers that came out on single cell, 3 or 4 use median normalization, 3 or 4 don't normalize across conditions. They use TPM, which will give you transcripts per million for a given cell, but it doesn't normalize across cells. And then other papers just homebrew normalization methods. Normalization has a huge effect, of course, and we find that it's a real problem. So we're working on normalization. That's all I'll say right now. But if you're analyzing single cell data, you should think about normalization because it's an issue. Okay. One way that you can check if you're working with single cell data if normalization works well is to look at the heat maps that I just showed you for all the quantiles. Look at them before and after normalization. So this kind of thing looks good, right? If I order, let's say you generate a plot where you order genes by median expression level on the y-axis, and you order them by library size on the x-axis, what you'll see prior to normalization is what? What do you see if this is normalized by library size on the x-axis? You go from what color to what color? Let's say your library sizes are really different. So you're going from like 2 million reads to 20 million reads. What happens? Expression increases, right? So 2 million reads, if you're kind of green down here, you have 20 million reads, you're increasing expression estimate prior to normalization simply because you have more reads, right? And normalization should get rid of that. So if you look at this plot prior to normalization, you should see some sort of green to red trend, and you often will, especially for the higher expressed genes. There's more variability in the lowly expressed genes, so oftentimes you just see a lot of variability and a lot of zeros. For the higher expressed genes, you'll see some green to red, and after you normalize, you should see that go away pretty much. But you won't in most cases, because normalization for bulk, the methods that work for bulk don't work for single cell. So that's a good check. And then what do? You do what we're doing, which is we don't know exactly yet. But we have a couple things on the go. But we know that we need to do something. So we're working on that. Yeah?
So I have one question. From my understanding of single cell, one of the important steps that you put in there is a preamplification step to actually amplify the amount of cDNA that's present that you're able to read. So you're really not doing the same technical approach that you do on bulk RNA-Seq, on single RNA-Seq. How much of that influences your ability to do normalization or your output
Yeah, so the question is, the technology to get single-cell RNA-seq data is different in a number of ways than with bulk RNA-seq, and the particular one that was talked about was with the amplification, that you need to, there's different amplification steps, and so there's amplification biases, which are different than in bulk. And how much of an effect does that have? It has an effect for sure. So what we're trying to do is write down a model that incorporates those biases to the extent that we can estimate them, and then, adjust for them. We know that it has an effect. The other huge effect, I'll tell you, is the number of zeros. So median normalization throws out any gene that has a 0 in it. So depending on the number of cells that you're trying to normalize, you can end up normalizing using 3 or 4% of your data. And that's not an extreme. I mean, for the single-cell data sets that we've looked at, we never see normalization. If we use median normalization, we never end up using more than 10% of the data to normalize, because there's a 0 in at least one of the cells for those genes. And so it ends up being a big issue. Now, there are some, we're looking at 31 single-cell data sets from the Thompson Lab and from some other collaborators and from publicly available. And I would say 15 of them look okay anyways. If you have pretty comparable sequencing depths and there's other things, median normalization will look okay. And one nice, the first step to be able to tell is to generate these plots. And if you see the library size, it's going to go, you're going to see an increase in expression if you order by library size prior to normalization. And if for most of the quantiles you see that go away, then you're kind of okay. And we see that. But for a lot of the data sets, we don't see that. And so that's an issue. Mm-hmm.
I'm a little bit surprised to hear you say that the library size is a critical parameter here, because a cell will typically only have 500,000 at most mRNAs. So if it's 0 with 500,000 reads, it doesn't really matter whether you go up to 50 million reads, it'll still be 0. In other words, if you correct for the number of times you're counting a single message, that should be pretty insensitive to the number of reads.
Yeah, I don't know that I think per cell, I understand your question. So the question is, you don't have that much expressing per single cell anyway. So if you go from 5 million to 50 million, you shouldn't see a huge difference. And if, I agree with that if you were looking at the same cell, but because from 5 million to 50 million, I think I see what you're trying to say. So hear me out and see if this makes sense. The capture efficiency per cell is so low that we get different mRNAs every single cell. I mean, the capture efficiency is between 10% and 40%. And that's not counting cell-to-cell heterogeneity. That's saying for the same cell, I'm thinking capture efficiency is 10 to 40%. So whatever mRNAs are expressing, I don't see the majority of them anyway. And now I'm going to introduce cell-to-cell heterogeneity. And so those are two huge issues. Does that make sense?
It helps.
Yeah. So are there other things about if you're working with single cell? Sorry not to give you some great advice on normalization, but we don't know what to do yet. Mm-hmm. Yeah. Yeah. So there is a nice approach. So the question is, have you thought about using unique molecular identifiers, or similarly spike-in? Spike-in's another thing that you can use to get your hands on what's going on in a cell. So we do have some spike-in data, and we do have some spike-in data, and we've thought about what to do with spike-ins. And there's a nice paper that just came out by Sylvia Richardson's group, if you have data with UMIs, on how to normalize. So if you have unique molecular identifiers, you have data with that. There is a normalization method out. We haven't had time to try it and see how well it works. So that's a real possibility. If you're spiking in, or if you're using that kind of approach, I assume it's going, let me say, if you're using that type of approach, I assume it's going to help. With the spike-ins, the people we work with in the lab kind of don't want to use spike-ins for all sorts of reasons that people in this room could probably tell me more than me. I mean, I know it sucks, they suck up a lot of reads, okay? But they're also kind of technically challenging from what I understand, so they would like a normalization approach that doesn't use spike-ins. So that's what we're working on. But yeah, those other, types of data, I think there would be some advantage. Yes?
I heard Sandrine Duagula talk about her work at Berkeley a couple of weeks ago at the complex trait community meeting. She said exactly the same thing you did, which scares the hell out of me. You have two really good statisticians telling you that after a couple of years they can't figure out what to do with the zeros. Those of us who aren't statisticians have some trouble.
Yeah.
The other thing she said is the spike-ins didn't work at all for her. And
Yeah.
it's just really hard to get good spike-in data.
Yeah.
But so sort of my idiot's question for you is, how many reads do you recommend for single cell? So it's all some people say 100,000 is enough if you're just trying to ID them.
Right.
The recommendation I've heard is don't go above 1 million. But then when Sandrine gave her presentation, she was going to 20 million. I said, why are you wasting all your reads?
Yeah. So that we have the same. We have from 500,000 to 20 million. And part of the reason for that is because we're trying to understand the technology, like how does normalization work and how does, variability change when we have more and more reads and things like that. So that's more a quality control thing. And I think just like with bulk, the number of reads you want is going to depend on what you want to see, right? So with bulk, they'll tell you, well, 1 million reads is more than sufficient if you want good estimates of expression for the 80% of the highest expressors. But if you want estimates of isoforms and you want estimates of, kind of weak, lowly expressed isoforms, then you need to go to much higher than that.
Doesn't make sense to me. We'll talk offline.
Okay.
It's not that much mRNA in the cell. So you either have it or you don't. You're going to nail it with 1 million reads.
Mm-hmm.
Is my opinion. But maybe there is an isoform issue I haven't thought of. So, especially as they get longer. So right now we're getting 200 nucleotide reads. You can imagine you have 500 nucleotide reads.
Mm-hmm.
Isoforms won't be an issue. And you'll know exactly whether you're reading from the same message before you can.
Yeah. Okay, I have to think about that for single cell more. Get my head around the amount of mRNA. I mean, I go back to capture efficiency so low, but that doesn't undo your point, right? That would even stress your point, like you're not going to see as much, so sequencing is not going to give you much more. Other comments? No, we're good? Yeah? No? Fixing your shirt? Okay. All right, so that's one thing, normalization, that we're working on. Second thing we're working on, as you might imagine, because we love differential expression, is how do you define differential expression in single-cell data? So this, we don't have anything submitted, but we're close on this, and I think we know how to do this better than we know how to do normalization. So with this, looking around for a board, I guess I'll just wave my hands and say, you can imagine if you have two multimodal distributions, one in condition 1 and one in condition 2, right? And so I'm just going to say they're bimodal. Like I have a bimodal distribution in condition 1 and a bimodal distribution in condition 2. And I'm not talking about zeros because you have, multimodality due to zeros too. I'm talking about the non-zeros. So what are all the things you might be interested in terms of differences? You might be interested in knowing if I have multimodality over here and multimodality over here, are they the same means? Right? I have mu1 and mu2 and mu3 and mu4. Are those means the same? If they are, is there differential proportion of cells? Like in condition 1, most of the cells are in the second mode, and in condition 2, most of the cells are in the first mode. Are the modes different? Are the first two the same and the second two are different? So there's all sorts of ways you can define differential expression. And we're developing a method where you can quantify posterior probabilities, much like EB-Seq, except the expression pattern options are much greater, right? There's not just different or not different, like there would be in a unimodal distributions within conditions, but there's differences due to differences in the modes, differences in the proportion of cells within each mode, and so on. So we're working on that. The last thing we're working on, I'll talk about today, which is identifying oscillating genes, okay? So as we know, microarray and bulk require thousands of cells, so you might miss oscillators because, just think about cell cycle. You're averaging out over cells in different phases of the cell cycle when you do a bulk experiment, but unless you synchronize, but not with single cell. You can synchronize, of course, if you want to get, estimates within bulk on expression dynamics in different phases of, let's say, cell cycle or circadian rhythms, but that'll change the dynamics of other systems. So if you synchronize for circadian rhythm, you might change cell cycle, and that's been shown to be the case. Single-cell RNA-Seq allows us to look in individual genes, individual cells, of course, but we can't do a time course in individual cells yet. So that would be cool if we could, but we can't. So our collaborators were interested in identifying genes that were oscillating, but they couldn't, in single cells, but they couldn't give us a time course. So they want us to do it from snapshot data. So how are we going to do that? So we developed an approach. It's coming out soon in Nature Methods, and we call it OSCOPE. So it's a pipeline for identifying oscillatory genes in unsynchronized single-cell RNA-Seq. And here's how it works. So this is what we imagine going on. We have here 4 cells, and I have 2 genes, and they're oscillating. Okay, they're in the same group, meaning they have the same frequency, but they might be phase shifted, and that's what I'm showing you here. These cells are phase shifted. The genes in these cells are phase shifted, but they have the same frequency. And I imagine them oscillating like this, but I don't see them oscillating over time. What I see is data collected at time t. All right, so I'm imagining this going on, but here's my data collected right here. Okay, now if I could order, if I knew, I don't know, but if I did, just go with me for a moment, by the lifetime of the oscillation, then I could reconstruct the oscillation pretty easily, right? So if I could order, I see this guy has been oscillating for the shortest amount of time, right, T4. It started oscillating here, and I collect data at time t, and I'm calling that the time of oscillation for cell 4. So there's T4. The second longest one is cell 1. That time's T1. If I knew this order, the length of the oscillations, and I could put the cells in that order, then I could reconstruct these cycles pretty easily. And I call one cycle of the oscillation the base cycle. You can see this base cycle repeats itself. So one cycle of the oscillation is called the base cycle. Now, I don't have that, right, because here's the data that I have. I have data collected at time t, so I don't know that cell 4 has been oscillating for a shorter amount of time than cell 1 or whatever. And if I just order them by collecting order, I don't see anything, right? It just looks like noise. So what I want to do is order them by what I'll call the cyclic order. I want to place these cells in an order that allows me to construct one cycle of their oscillation called the base cycle. So can I reconstruct the cyclic order? And the answer is yes, but I want to make sure you know what cyclic order is before I go on. So let me just re-review this. Here's the data that I have, and I want to put these cells in an order so that I can reconstruct one cycle of this oscillation called the base cycle. Now, why can't I get both cycles? Well, you can see, like, this data point here, T4, in terms of what I see, looks the same as T3. No, I'm sorry, T2. So here's T4, the red's there and the black's there, and here's T2. Sorry. Cell 4 at time t, here's gene 2 and gene 1, and here's gene 2 and G1. So the data at time t for these 2 cells looks the same. So I can't tell that, oh, cell 4 is over here and cell 2 is over here. I don't know that. But if I can order cell 2 and cell 4 very closely, I can reconstruct one cycle of this base cycle. So that's my goal, is to order these cells, define their cyclic order, which is their order along one cycle of the oscillation, which I call the base cycle. Does that make sense, kind of? I screwed up cell and gene for a while, so I'm going to pause extra long so that you know what's going on. Okay. So how do we do that? Is there any hope of doing that? There's hope, right? because we have a method now. All right, so here's what it looks like. So step one is I want to find, out of all 20,000 genes, I want to find some candidate oscillators. Now what's cool is we don't know what's going on in terms of order, but if I plot one oscillator versus another, and they have the same frequency, but they're phase shifted, what kind of shape will I see? Yeah, a loop or an ellipse. That gives us a lot of power, right? Because we can scan all pairs and we can group genes by a measure of how well they fit, in terms of gene pairs, how well they fit in an ellipse. So again, if I have a collection of genes and they're oscillating with the same frequency but they're phase shifted, and I plot one gene versus another, I see an ellipse. So by looking at phase plots from all pairs of genes and fitting an ellipse to them, I get a distance measure. How far is this pair from an ellipse? And if they're really close, they end up going as candidates in the same oscillatory group, we'll call them. So we pull out in phase one, we screen for potential oscillators just by looking at phase plots of gene versus gene and fitting an ellipse to them. Okay. Then once we get these candidate oscillators, we cluster them into groups that likely have the same frequency, but they're phase shifted. And we just do that by the score that we have after fitting an ellipse to these genes. Then the tricky part, once we have a group, is we need to reconstruct the order, right? So how are we going to reconstruct that cyclic order? How do we order cells so that we can reconstruct one cycle of their oscillation called the base cycle? And so what we do is we borrow, actually extend an algorithm that was developed 30-some years ago to deal with the traveling salesman problem, and it's called the nearest insertion algorithm. So in the nearest insertion algorithm, what it does for the traveling salesman problem, so remember the traveling salesman problem, you have a list of cities, and you only know the distance between each pair of cities. That's what you have. And you want to find a route that takes a salesman from one city, visits every city exactly once, and returns to the origin city, and you want one that minimizes the distance. That's your problem in the traveling salesman problem. And a bunch of algorithms have been developed to address this problem, and one of them that's shown to work really well over the decades is called the nearest insertion algorithm. So in terms of the nearest insertion algorithm, what happens is you start off with a triple. You say, okay, suppose these are, well, you can actually start out with a double. That's even simpler. Start out with a double, pull in the third, and ask, where should I place this third city? In between the two or on each end of the other one? Calculate distances and repeat. And you keep iterating through these options. And there's sort of efficient programming algorithms that let you iterate through these options till you find an optimal solution. So in our case, the problem is really similar. For each gene cluster, we want to find the optimal route through the cells, right? And in a cool way, we want to return to the origin cell, because even though I'm showing these as a time course, this is cyclic, right? It's returning to the base cycle. So we want this thing to be in a loop, as you would be in any kind of cyclic pattern. So we want to order cells in such a way that we minimize some kind of distance. And the extension to the nearest insertion algorithm is how we define that distance. So we want to order cells in such a way that we minimize distance between the gene expression pattern and the baseline oscillation of that group. All right, I'm going to pause again to see, because that's like the whole thing of the method. We identify, yep.
All these genes have the same amplitude, like you normalize them?
That's a great question. Do all the genes have the same amplitude? We normalize them so they do. Yep. We have to scale the genes so they have the same amplitude.
How do you get the frequency from the ellipse?
We don't. How do you get frequency from the ellipse? All we know is if they're oscillating with the same frequency but different phases, then the ellipse will fit well. So then we pull out candidates by looking at how well pairs fit in an ellipse, and we don't get frequency from that. Yeah. Okay. Maybe some results will make you happier. All right. So here's some results. So does this thing work? We think it does. So the first thing we do is we look at microarray data, because this was a microarray time course where they had a pretty large number of data, and they had gold standard testing on which genes were oscillating, like they did other experiments to try to validate which genes were oscillating. So we call this the Whitfield data because Whitfield's the first author. They did a time course experiment on HeLa cells. They were synchronized for cell cycle. They have 48 samples. One was taken every hour, which is about 3 cell cycles. So we permute the data. We pretend that we don't know the order, right? And then we apply OSCOPE and we see how well it does. So we permute the data and we identify some clusters, which again are groups of genes that are oscillating with, we think, similar frequencies, but possibly different phases. In fact, they have to have different phases. Anybody cares about that, I can tell you later. But they're oscillating with similar frequencies that are phase shifted, and one of the clusters had 69 genes, and we find that 65 of the 69 were validated as oscillating in the Whitfield data. So here is the known time course order on the right for 4 of these genes. The 69 genes are in the supplement of our paper, but I'll show you 4. So here are 4. This is known time course order. And then here's our extended nearest insertion algorithm reconstructed order. And again, we can't figure out where you are in terms of how many oscillations have taken place. All we can do is reconstruct one base cycle, right? One cycle of the oscillation. So we order cells in such a way that the base cycle is reconstructed. And we can see that we also can estimate pretty well the phase shift within the group. So you can see here, I've marked on the known time course order where the highest peak is, and I mark that similarly in our reconstructed order. So we get a good estimate of the base cycle, and we get a good estimate of phase shift for these genes. So it's 1 sample every hour, and it's done for 2 days. Yeah. Yeah, so the question is, how many cells do you need? The lowest we have done is probably this 48. So we have some data. I don't know exactly how low you can go, but I don't think you can go much lower than 48, which is not usually a problem because the C1 platform gives you 96 cells, and usually 90 or so are good. But you know, in terms of reconstructing order, I'm going to say something like 48 is as low as I would go at this point. We're looking a little bit, we've tried to go to 24, we can get some stuff with 24. So John Hoganesh recently published a PNAS paper that gives you like atlas of circadian gene expression across mammals, right? And he looked at circadian gene expression in all sorts of brain, muscle, all sorts of tissues. And we've looked at, and they have like 96 samples. So we've downsampled to see how well we can do. Because another thing OSCOPE can give you is, even if you're not interested in reconstructing order, it can tell you an estimate. It gives you an estimate of how many genes are oscillating. So for stuff like John wants to do atlas of circadian gene expression in the brain, and he wants to look at 20 brain regions or something, but he doesn't have enough money to do 20 brain regions. So he would like us to tell him, if he does one experiment from each of the 20, which 10 regions to look at with time course, right? So we've been trying to figure out how much data we need in that initial experiment to get good estimates of the number of oscillators, and I don't think we can go super low. Yep.
So when you have the gene cluster, you can order the cells based on these distance measures. I think you mentioned about like how to measure the gene, just how do you get gene clusters first? Like by measuring like what's the distance they got?
Yeah. So the question is, how do you get the gene clusters first? Like I talked about within cluster how you get the order of the cells, right? But how do you even get a cluster to try to determine order? And I need to put a slide in on that, so my apologies, because I'm kind of hand waving up here. The way we do it is we start out with all the genes, and we actually do some pre-filtering, but you don't need to if you want to let your computer run for a long time. And we calculate for every pair of genes, we fit an ellipse, and we calculate the residuals from the best fitting ellipse. Okay, so now for every pair, I have residuals from that fit. I can calculate goodness of fit, like the sum of the squared error of the best fitting ellipse to that pair, right? Where those residuals are very small, those are the genes that are oscillating with the same frequency but perhaps varying phases. So that's actually, that sum of squared error is actually the distance measure that we use in a clustering algorithm to give us clusters of genes. Does that make sense? Yeah. Okay. Okay, here's some more results, H1 cells. So we applied OSCOPE to 213 human embryonic stem cells, and we found a 29 gene group that looked like cell cycle, because 21 of the genes that we found were annotated as belonging to cell cycle in Go. So to investigate this group, the Thompson lab did another experiment where they took 247 human embryonic stem cells and then they labeled them with this fluorescent, I don't know, everybody can tell me what Fuji is, but I can't remember what Fuji is. It's a label where it's a color label where you can tell which phase of the cell cycle the cell is in, right? So now we have 213 unlabeled and 247 Fuji labeled cells. And here's what they look like. So in color, I can see which ones are in G1 and G2 and S and so on. And then we pretend we don't know that color assignment, right? And we apply OSCOPE again, the extended nearest insertion algorithm part of OSCOPE, on the 29 genes that we found. And if our ordering has something to do with cell cycle, we should see that in the coloring, right? So here's what we find. These are 4 of the genes, again, 29 are in the paper if you want to see them, and we can see that the Fuji labeled cells, the ordering is consistent with cell cycle phasing for the most part. Okay. The other thing we found with OSCOPE was, again, Ning came to my office. I cried when Ning left because she was so good. But she works in Jamie's lab now, so I still get to see her a lot. She came in with what she calls the Smiley face pattern. So we found, we applied OSCOPE on these human embryonic stem cells, and we found some interesting clusters. I told you about one. I thought, well, we better find cell cycle, and we did, so that was fine and nice. But then she found this pretty big group, and we had no idea what it was, and she called it Smiley face for the obvious reasons. So we're like, what is this Smiley face about? And she got her sleeping bag and stayed in the lab for many days because I figured it would be good to follow this protocol through step by step, right? Because as a biostatistician, I have a rough idea of what goes on, but I don't do the stuff and I don't have a super good idea of what goes on. And I figured there must be something strange because we had checked, of course, batch and all these things that statisticians like to check, and it wasn't associated. The Smiley wasn't associated with anything obvious like that. So she figured out that it's actually associated with the position on the C1 auto-prep array. So if you've done this experiment, you've seen this thing. This is, C1's auto-prep array and their IFC system. You load cells in somewhere, I don't know, somewhere around. Somebody show me where you stick cells in there, and then it isolates this magical system, as far as I'm concerned, some sort of magic happens, and it isolates 96 cells, right? And then they can be visualized, so you make sure they're good-looking cells and they're not dead and things like that, and they're killed in there, and then amplification happens in there, and all sorts of things. But here's where the 96 cells are isolated. It's this 16 by 3 grid here and 16 by 3 grid here. And then you pull the cells out of here for sequencing, and you're supposed to pull them in a particular order according to the C1 manual. And what she's found is that there's increased expression, not in every single experiment we've ever seen, but in many of them, there's increased expression based on where you are on this system. So if she colors cells that came out of these 2 by green, you see this. We see increased expression for these. This is H1 cells that we first saw the Smiley face pattern. We don't call it Smiley face pattern in the paper, but that's what we call it offline. We call it ordering effect in the paper. Then our collaborators did some experiments to make sure this wasn't kind of downstream, so they did RT-PCR and sequencing off of mRNA that came straight out of the C1 system, and I'm only showing you 2 genes, but they've done a few more that are in the paper that you can look at. Not a ton, but a few more, so we're pretty sure it's coming off this system and not something downstream. This isn't publicly available data for these genes. This is another publicly available data set where we see the same kind of thing. This is another summary of that. So in panel A, we see our H1 Fuji data. Panel B, data from Trapnell's paper, Quake Lab paper in panel C. And then we talked to Fluidigm about this. They don't think it's an issue, I think. They sent us this, and I see it's less of an issue in data that they have in-house, but we're still pretty convinced there's something at least to check for going on here. What's that? You know, so the question is, did Fluidigm say they've never seen it? So our collaborator, the Thompson Lab, works with the Fluidigm reps, and they looked at, we showed them this before we sent it out, and they looked at data, and they say they don't see it there, which is, you know, arguable. I mean, we see it a little bit. You know, this one's pretty clear, I think. But you see it less in their data, but we see it pretty strongly in these other data sets. So I would say I think it's something to check for. You're not going to see it in every data set, but you're going to see it in some data sets. So we think, and we see it in a lot of genes. You know, you're not going to find it by chance. Yeah, yeah.
Why would that be happening? Like, what, I'm trying to
Yeah.
kind of understand, you know, I don't know why.
Right, why? I have no idea. We don't know. So I don't know. Maybe a biologist here has an idea. I would love to hear it. We have tried to figure out what's going on. There's something with the sequence of these genes. Is there something with, you know, if you look closely at the autoprep system, like these cells have to turn around the corner? Like, is there something with the speed with which they make it through this platform? I just don't know. I don't know. Why? Stress, yeah. Shear stress, yeah. I don't know. You know, because it's on both ends, right? But it's on both ends. Like, I don't know. Yeah, there's channels there, so it might be shear stress of them turning the corner. I don't know. Mm-hmm.
Yeah, I run a technology core at UCLA, and I have no doubt that there is a Smiley effect. I have a long history of companies running all of their tests on perfect DNA and perfect cells and perfect data. So I'm sure that they have something that they've trained that they use for their sample that doesn't have any problems. It's very typical the way these companies.
Yeah. Okay, so I'll tell you the preview. Is anybody interested in methods for identifying somatic mutations in cancer? Here are the options. I can summarize here, and we'll come back and look at some code for EB-Seq, and then I'll give you a little easy ovarian cancer story. Or we can take 10 minutes and talk about methods for somatic mutations in cancer, and then do shorter ovarian cancer story. Anybody care? Well, we're still going to we're doing our code no matter what, but what's that? Let's learn about somatic mutations? Yeah, okay, I'll make it quick. This is more of an advertisement. It's called Magic. My former student, Keegan Korthauer, is really good at naming stuff. So if you're interested in identifying driver mutations in cancer, how do you do it? This, again, I tried to convince this student, do not work on this problem when she came to me, because there's a gazillion methods out there, and, you know, what are you going to do? Make one method a little better maybe in one condition? Like, I was super negative about it. And she came back and told me she wants to work on it and told me why, and I'll tell you why it ended up being a good idea. So I reluctantly advised her. Now I'm happy I did. Anyway. So methods that are out there that a lot of people use, they're frequency-based, and they say, okay, here I've got some genes, here I've got some samples, I've got a black thing here, black dot here. If this gene is mutated in this sample, that's my data. And I'm just going to say you're a driver if you're mutated a lot. And a lot means significantly over some background mutation rate that's calculated across the genome. So these are called frequency-based approaches, or we call them frequency-based approaches because they're based entirely on how frequently a gene is mutated across samples. And so like P53 shows up with those because it's mutated all the time in cancer, and some other stuff shows up. But it's a little bit tricky because background mutation rate varies a lot across the genome, and so the trick with frequency-based methods is getting a good model for background mutation rate. Back in the early days, people used a constant background mutation rate, and they started finding all sorts of olfactory receptors. And then Eric Lander gave a talk in an AACR meeting, and he said, something smells fishy. And what's going on is that these olfactory receptors, they're just, I don't know why, but they mutate a lot, right, for whatever reason. And so they would be highly mutated relative to a constant rate across the genome. So you have to adjust for these types of things, or else you find things like olfactory genes as being drivers of cancer. Okay, so the trick there is to get a good model for the background mutation rate, not just a constant across the genome. Then there's these functional impact-based methods where they take into account, they have some sort of measure of functional impact of a mutation, maybe SIFT, sorting intolerant from tolerant or whatever. They think about this particular mutation, how likely is it to have functional impact on the downstream protein? And they use that as a measure that tells you how likely the gene is to be a driver, right? And then finally, there's at least one method that looks at positional information. So where do the observed methods cluster, right? So in passengers, they're more kind of sorted out at random, these mutations, whereas in drivers, they're more clustered in important positions. But the problem with the positional-based clustering is that you don't often see this kind of thing. You don't see these mutations piling up in a given region. So on the left-hand side, what I'm showing you are data from the Cancer Genome Atlas Project, ovarian cancer study, which I thought we'd be talking about at 4:15 yesterday, so I thought you'd already know what this is. I'll say briefly that TCGA is, in case you don't know, a cool project, started in 2001 by the NIH. When they had money, they wanted to think about what to spend it on. They thought, okay, let's try to do something with cancers. And ovarian was at the top of the list because it's one of the most difficult cancers, meaning diagnosis is often late and prognosis is often not good, and lung and brain were the other two. And the idea was to collect relatively large groups of patients, like 500 or so from each cancer, and all the genomic information that they could get their hands on, put it in the public domain, give some data analysis groups money to analyze that data and try to get some insight into the genomic basis, further insights in the genomic basis of these diseases, and importantly, into how they might respond to various treatments. So with TCGA ovary, we have about 500 patients. We have somatic mutation information for those patients. And here I'm showing you for some pretty popular genes, KRAS and so on. Here's amino acid position along those genes. And you can see this is, there's a little bit of clustering for mutations in KRAS here. But we don't see a ton of this piling up, right? But if you go to COSMIC, so COSMIC, the Catalog of Somatic Mutations in Cancer, which is cataloging cancers and somatic mutations in cancers across thousands of cancers, independent of type, right? Then you see a lot of this piling up, right? You see a lot of positional clustering of mutations. And so my graduate student really wanted to use this data. She wanted to combine frequency approaches with functional impact information, because no one had done that yet in one model-based approach, and also use data from Cosmic in priors so that we could get a better idea of the driver mutations in cancer. And I kind of thought, okay, maybe this is an okay idea, but I still didn't think it was going to matter that much, but she showed me that it did. Okay. So MAGIC, what does it do? Again, frequency of mutation, it improves the background mutation model. I told you tricky part with frequency-based approaches is you need a good background mutation model. And it in a way that adjusts for gene-specific factors like replication, timing, and expression level. So John Stam, just call him that because his name Stamatoyanopoulos is hard to say, showed us years ago that replication timing has a real effect on mutation rate, and you need to adjust for that, right? Or you start identifying genes that are in the late, I think it's, where do you have more mutations? In the late stage, because there's not enough time in the cell cycle to take care of those, repair those mutations, and so there's a bias in terms of the genes you identify with respect to replication timing. She adjusts for that. And also expression level. She incorporates functional impact with SIF scores, although you could use something else. She gets the prior probability, as I mentioned, of driver gene status from Cosmic. And when we fit, when we try to say something about ovarian cancer, we don't use ovarian cancer data from Cosmic, but we use the other data, not to bias ourselves. And what you get at the end of the day is a ranking of genes based on posterior probability of being a driver. So in summary of everything I've told you this morning, and I can talk more about MAGIC. I told you it was more of an advertisement if you're interested in identifying driver genes in cancer, so there's not a lot of detail there. But the things to remember from this morning, I think methods for identifying DE genes in an RNA-Seq experiment aren't super great if you're interested in identifying DE isoforms, so we developed EBSeq. And EBSeq HMM if you're interested in ordered experiments. And then OSCOPE, this is pipeline for identifying oscillatory genes in single-cell RNA-Seq. Even if you're not particularly interested in oscillatory genes, you might be interested in adjusting for their effects. If you're interested in some other inference, right? But you've got cell cycle oscillations affecting your data. You can identify them, estimate the base cycle, regress it out, so you can adjust for the effects of cell cycle. It'll give you more power to figure out, to address the questions you're trying to address in the single cell experiment. That's part 2 of this, but I haven't shown you any results of that, but I have a good hunch that that's going to be important in terms of improving power in single cell experiments. And then MAGIC is what I just talked about. In terms of availability, and again, I'll be showing you some code, hopefully, after the break. EBSeq's in Bioconductor, it's on Galaxy, and there's a graphical user interface that allows you to upload expression estimates in a CSV file and get your posterior probabilities back in a CSV file if you don't like to use R or whatever. You can use that. EBSeq HMM is also in Bioconductor. OSCOPE is, the package right now is just at my website. We're going to get that up to Bioconductor soon. This is coming out soon. And we're working on just a quick note and package called OE Finder, where you can check for ordering effects in your own single cell data. So maybe if you're using data from Fluidigm, you won't find any, but as we've heard, we see it a lot, so you might want to check in your own data. And then if you like MAGIC, that's at my website too. So thank my collaborators. Li Fong has been invaluable in the single cell work. He knows everything about single cell. Other members of the Thompson lab that we've been working with for years on EBSeq and EBSeq HMM and other things. And I'll thank you guys again for your time and attention. Happy to take questions, and hopefully we can get some code going for after the break. Thanks.
Doing EPC for single cells differential expression analysis, should we have to take care of oscillation patterns into account, because does that lead to more of a multimodal distribution, or if you
Yeah, so the question is, do we have to like use O-scope to accommodate for oscillations before applying EB-seq with single cell data? And a couple of things. I do think using O-scope to adjust for oscillations for some of the genes prior to downstream analysis will be important. There's a paper, I'm trying to think of I can't remember the first author, I can tell you after the break, that came out in Nature Methods within the last 6 months that adjusts for oscillations in cell cycle, and they show that all sorts of genes that they didn't know, that they expected to be involved in immune response, they didn't find prior to adjusting. They adjust for cell cycle, and they're very clear. So based on that work, I think adjusting for the oscillations in single cell data prior to downstream analysis is going to be important. That's point one. Point two is I wouldn't use EB-seq. We do use EB-seq now to look for differential expression, but it's pretty bad idea because for a lot of the genes, the model doesn't fit well because it assumes things are unimodal. And so soonish, we will have an approach that's EB-seq-like. It's a mixture model-based approach that accommodates the multimodality and also allows you to address kind of more detailed questions, not just different or not, but like different in what ways. Yeah. Steve? Yes. So the question is, can you use O-scope not just for single cell, but for bulk, like if you're looking for circadian rhythms? And yes, the answer is for sure. So this is why we hooked up with John Hoganesh and started analyzing his data to see if we could find circadian oscillations in these tissues. One, because he was interested in experimental design. Like, can you tell me where to look in brain that has the most oscillators? And two, we were curious exactly for the reason, can this be useful in bulk? And so I think it can, yeah.
So I have a very theoretical question is, when you feed the ellipse model, what about the highly correlated genes that this is a diagonal and how you fix the ellipse model the redefined phase?
Yeah, this is a great question. So how does this ellipse thing work when you have perfectly correlated genes? Then the trivial ellipse fits perfectly well, and you'll pull them out, right, because they'll have zero residual. And this, we weren't smart enough to realize this a priori, but we saw it when we did it, right? So we ended up with groups that we thought these should be oscillating, but they're just super highly correlated, and they don't look like they're oscillating at all. So we have now, we have to restrict the parameters in the fitting of the ellipse so that we don't fit the trivial ellipse. And that takes care of that. But yeah, that's a great question. I wasn't clever enough to figure that out before we did it.